in the topic of ECM.
नहीं उठाई, बहुत परेशान हुए। हम कहे कि लगाए बसंती जो है ना, तो हम कहे कि भैया ऐसा है कि आपके परिवार बोल
Hi dear students, in this video we will be talking about extracellular matrix. In the topic of ECM, right, one of the topics like collagen is one of the very important question, right, that can be asked in exams very frequently. So let's talk about it. So what do you mean by extracellular matrix? So guys, anything which is out of the cell, right? So when the cells are being surrounded by certain compounds, certain materials, that is called as extracellular matrix. It has a protective role and it is also providing stretchability and some other communication purposes, right? So making it stable, right, and also helping in certain other roles. So anything which surrounds a cell is extracellular matrix. It is out of the cell, basically. Point is that. What it is made of? So guys, it has various structures. One of them is proteoglycans, right? Then we have certain proteins which are called as fibronectin and laminin. Then collagen, elastin, these are structural proteins, and also fibrillin. Collagen, elastin and fibrillin, these are structural proteins are present. Okay, fibronectin and laminin are specialized proteins that we'll talk. Okay, now out of them, let's talk about first of all collagen. So guys, collagen is the most abundant protein in the human beings. First thing, there are various forms of collagen found in humans, right? But out of them, few we must remember, like type 1 collagen is mostly found in the bone. As you can see, bone is ending with 1, B-O-N-E, 1, right? So it is found there. Then type 1 collagen is also found in cornea. If you see clearly, cornea is also having 1, though the R is there, right? Then, guys, this is found in the fibrocartilage. Okay. Type 2 collagen is found in hyaline cartilage. Type 3 collagen is found in skin and the blood vessels. Okay, and type 4 collagen is found in basement membrane. You just remember 4 and the floor. So floor is like basement membrane. Okay, like the basement membrane of kidney, if I say, or the cells, any cells. So guys, these are the 4 common names that you should know, and their location you should know. Okay, so bone is type 1, especially the bone is very important here. Now, moving to our next point. Guys, collagen has a structure, a very special secondary structure known as triple helix. Okay, so it has 3 alpha chains. Now, see, guys, I'm saying alpha chains, I'm not saying alpha helix. Okay, these chains have been named as alpha chains. So, collagen has 3 alpha chains, and these are having left-handed helical pattern with the 3.3 amino acids per turn. So alpha helix was different. It was a right-handed helix, right, with 3.6 amino acids per turn. So just ignore it right now. But you should know, if the examiner asks you that alpha chains and alpha helix, do you know the difference? Yes, alpha chains are just chains of the collagen present in helical pattern, right, in the collagen. So now repeating again, collagen is the most abundant protein in human body. It has a specialized secondary structure called as triple helix. Okay, triple helix. This triple helix has 3 alpha chains. Each chain, guys, has 3 amino acid repeat. That is called as glycine XY repeats. Okay, so there are around 1000 amino acid in one chain, So that means glycine XY is repeated 333 times on average, right? So first thing here is that glycine is the most common amino acid. As you can see, it is every third amino acid is glycine. So 30% of the collagen is actually made of glycine. So why so much of glycine is found in collagen? Guys, when the chains are being twisted, it's a triple helix now. So when the chains are being twisted, imagine the structure of rope, which is having 3 chains. So when you are twisting the chain, at the end of each twist, there is very little space. And this little space can be easily occupied by glycine. Why? Because glycine has a very small structure, the smallest amino acid, isn't it? So, guys, remember, glycine is the smallest amino acid, and that's why glycine provides flexibility to the protein structure. So my question is, which amino acid provides flexibility to the protein structure? Answer is glycine. Okay. Now, guys, X and Y. So the most common amino acid at the position X is proline, and the most common amino acid at position Y is hydroxyproline, right? But this can be occupied by any other amino acid, commonly by proline and hydroxyproline. So guys, why there is so much of proline present in the collagen structure? So guys, if you remember the structure of the proline, it was having a ring, okay, right? And that ring has a property that it doesn't allow the formation of alpha helix. If you remember, I told you that proline is a helix breaker, right? So it doesn't allow the normally formation of alpha helix, but guys, it inserts a kink or a twist in the structure. So this is what I want here, that is twisting of the structure, isn't it? So we want a structure which is twisted, okay, and very strong. So that is because of the presence of proline. These twist or the helical formation was being allowed, okay, with the sharp turns. So just remember one line: proline allows the sharp turning or folding of the proteins. That's why a lot of proline is present in the triple helix of collagen. Fine, guys. Okay, hydroxyproline we'll talk later, how it is formed and what is the importance. So that is the basic structure of collagen, guys. Fine. Now, how the collagen is synthesized, right? Guys, collagen is just like a normal protein, and it is made by the ribosomes, which are present in the cytoplasm initially. But it has some sequences known as signal peptides or the leader sequences, which will transfer the ribosomes present in the cytoplasm to the rough endoplasmic reticulum, right, where the completion of the protein synthesis will happen. So guys, once the collagen chains are completely made, now we will transfer them to the lumen of endoplasmic reticulum, where there will be certain modifications done. So the very important topic now we'll talk is the post-translational modification of collagen. So guys, the post-translational modification of collagen will be divided into 2 parts: intracellular, inside the cell; extracellular, out of the cell. Now inside the cell, guys, most of the proteins are modified in endoplasmic reticulum or the Golgi apparatus. So again, the site is same here. So we will do all the intracellular modification. The site is endoplasmic reticulum. Okay. Now what we have to do here? The number 1. The first modification, guys, is hydroxylation of proline and lysine residue. Guys, remember, lysine concentration or the percentage in collagen is very less, right? It's almost 1% only. But this lysine has been chosen as a site of sugar attachment and also hydroxylation. So that's why we have to do the hydroxylation at lysine also. So let's see how. So first of all, number 1 is hydroxylation. So we will take the enzyme proline hydroxylase, which converts proline to hydroxyproline. And we will take the enzyme lysyl hydroxylase, which will convert lysine to hydroxylysine. Guys, the enzymes which are doing this hydroxylation reaction, they are vitamin C dependent. Actually, vitamin C is helping the iron of these enzymes to be present in the ferrous state or reduced state. So that is the job of vitamin C actually, to make the iron in reduced state. So vitamin C is very important, required coenzyme for this particular hydroxylation reaction. And that is why, guys, the deficiency of vitamin C can lead to, weakening of the collagen, right? And scurvy with increased bleeding tendencies. Okay, fine. In the scurvy, guys, what you see normally, you will see the bleeding gums and the bleeding under the skin, right, or around the hair follicle, that is called as perifollicular hemorrhages, right? Follicle, hair follicle, and around the hair follicle, perifollicular hemorrhages. Bleeding gums can also be seen, and the bleeding can happen in the joints or in the bones also in the extreme cases. This can lead to the pain in the bones of the baby, and the baby will prefer not to walk but to keep their bones folded. And baby looks like having paralysis, and sometimes we call it as a pseudo paralysis. Okay, so baby folds keep the hands and the legs folded in a position which relieves the pain. So that is called as pseudo paralysis basically. That we'll discuss again in the vitamin C when we discuss. Okay, fine. Now, so hydroxylation is done. The second important point here is that, guys, why the hydroxylation was done actually. See, hydroxyproline is the site of hydrogen bond formation between these chains, and that is making the collagen a strong structure, stable structure, right? Everything counts, guys. This may be the weak bond, but yes, it is helping in the initial stabilization of the collagen, right? Then second modification now after hydroxylation is glycosylation, sugar attachment. So guys, glucose or a disaccharide known as glucose galactose together, right? So two sugars can be attached at the oxygen of hydroxylysine, OH of hydroxylysine, I mean to say, right? So guys, that's a kind of O glycosylation. We will see in some other chapters that there are two types of glycosylation: O-linked glycosylation and N-linked glycosylation. So O-linked glycosylation happens on the OH group of certain amino acids like serine, threonine, tyrosine, and now hydroxylysine. So guys, collagen is a O-linked glycoprotein, sugar containing protein, glycoprotein. So O-linked, remember this. Okay, so now we did. So why hydroxyproline is required? Hydroxylysine is required. Reason is clear now. What is the job of glycosylation, guys? It is not very well understood, but yes, it is definitely important because once you remove the sugar from the collagen, it is unstable. So definitely it has some role in stability, which is still poorly understood. Okay, now moving to the next point. So guys, these two are done. Third point is, guys, collagen, at the ends of the collagen chains, there are C terminal and N terminal peptides are present. Guys, these peptides have a lot of cysteine, right? So there is a alpha chain. At the ends of alpha chain, there is a lot of cysteines in both the sides, N terminal and C terminal. So guys, these cysteine residues, bind with each other and make the intra-chain disulfide bonds. As you know, the disulfide bonds are formed between cysteine chains. So if I take three chains, right, and they have the cysteine residues, which will bind with each other and make the disulfide bonds. So that's how, guys, they will make a triple helix. So already we have made a triple helix because of these disulfide bond formation at the ends, right? Okay. So these are called as propeptides. So the ends of the collagen which has been made now is called as propeptides, N-terminal, C-terminal propeptides that we will remove out of cell. But before that, now everything is ready. Collagen has been already converted into a triple helix, right? Okay. And now from the ER lumen, they will enter into the Golgi apparatus, and the Golgi vesicles will fuse with plasma membrane, and the collagen will be delivered out of the cell. Right, guys? So now the next part of our topic is post-translational modification, which are extracellular. We discussed intracellular. So in the extracellular area, what we have to do now? First of all, number one, we will remove the N and C terminal propeptides, okay, which were having a lot of cysteine, with the help of the enzymes which are called as propeptidase. So guys, these will be removed. Okay, now what we are left with is a triple helical structure of the collagen. Now the second step, guys, in the second step we will try to make a structure of the collagen where each triple helix is binding with the other triple helix, okay, in such a way that 1/4 of each chain is overlapping the 1/4 of other fiber. I will not say the chain word here now. We should use the word fiber now because it's a triple helix now. Already a triple helix is formed. This is called as one collagen fiber. And this is the second collagen fiber. Okay, so there are a lot of collagen fibers are out right now, out of the cell. So they will bind in such a way that there is one fiber is overlapping 1/4 of other fiber. So guys, you must highlight this line, and this is called as quarter, means 1/4. Quarter staggered arrangement of the collagen, like especially the bone collagen, for example, if I say. So quarter staggered arrangement of collagen is very important to write in the exams and remember in the for Viva. Fine guys, how the fibers are making bonds with each other, let's understand this. So guys, as we know, collagen has certain lysine residues. Okay, few were already hydroxylated, but there are few which are still not hydroxylated. So these lysine residues, guys, they will be acted upon by a copper containing enzyme known as lysyl oxidase. So lysyl oxidase will convert lysine to aldehyde lysine or oxidized lysine. We call it as a short as L-lysine, L-lysine. So guys, this L-lysine is formed now. And this aldehyde lysine or oxidized lysine is very reactive. So one fiber which has now aldehyde lysine will bind to the other fiber, at the same lysine residues, which is unmodified. So guys, this is called as aldol condensation. Okay, so these oxidized lysines, they are reacting, and the reaction is called as aldol condensation reaction, and this makes the collagen very strong structure. Right guys, so what is the bond present? Aldol condensation is the name of the reaction that is happening between two fibers of collagen. Correct guys, that makes the collagen a very strong structure. Fine. So what was the name of enzyme? Lysyl oxidase. It contains copper. Fine. And guys, that is why the deficiency of copper, can cause a disease which affects the collagen. So what's the name of that disease? Menkes disease. Menke. Menkes disease. Right? Menkes disease is caused by deficiency of the copper, and this may present with the weakness of the collagen. Right, and this leads to weak collagen of the blood vessels, which may rupture and cause death. Right, and there are certain other symptoms that we will discuss in minerals. So refer to the copper deficiency in the mineral section. Okay, so that collagen is being completely formed and ready, right, to make the bones. Let's say now there are certain disorders, guys, in the collagen that we must at least remember the names. So what are the disorders of collagen? So guys, that's how the collagen is being completely formed, and it's ready, right, to make the bones, let's say. Now there are certain disorders, guys, in the collagen that we must at least remember the names. So what are the disorders of collagen production? Yeah, collagenopathies. So like number one, scurvy, we have discussed already. Number two, Menkes disease. I told you it's a deficiency of copper, but yes, affecting collagen, so it's a collagen disorder. Then, guys, we have a disease called as Alport syndrome. Alport syndrome affects the type 4 collagen, right? And the basement membrane of the kidney is affected, and this leads to renal failure and blood in the urine, hematuria. Okay, so this causes hematuria and progressive renal failure. So that is Alport syndrome. Then, guys, we have a disease which is called as osteogenesis imperfecta. See, guys, what happens is bones, osteo means bones, and imperfecta means not formed properly. So the babies who are born with osteogenesis imperfecta, they have a mutation which leads to replacement of the glycine with some other bigger amino acid. Let's say the big amino acid is, if I say tyrosine. So glycine, which was the smallest amino acid, is now replaced with tyrosine, which is a big amino acid. So this will destroy the structure of collagen. The triple helices will not form properly, and this causes the weakness of collagen. And guys, if the collagen is weak in the bone, let's say, what will happen? They will just, break easily. Okay, so bones in the osteogenesis imperfecta breaks even at the minor trauma. Okay, and the babies or the adolescents or the adults or the childs, they all present with the broken bones, multiple fractures, right? So multiple fractures. See, guys, collagen is flexible. Glycine makes the flexible proteins, but if glycine is replaced by other amino acid, so the flexibility will be lost. As we say now that children have their very flexible bones, they don't get fracture easily, right? But these babies, they get easily, even at the slightest trauma. So that is osteogenesis imperfecta, and one of the feature of osteogenesis imperfecta is also there is the blue sclera. Blue sclera. That's not white; it's bluish. Fine, guys. Okay, so that is one more disease that we have discussed. Then, guys, some more names. One is Ehlers-Danlos syndrome. In the Ehlers-Danlos syndrome, guys, what happens is, there is collagen type 1 and collagen type 5 are affected, and the skin is hyper stretchable, hyper elastic skin is there, and the joints are hypermobile. You might have seen these people on the TV that they can just take their skin out away from their body, like from the chin to the chest, right, like that, and they can move their joints completely, right? Okay, so that is called as a hypermobile joints. Fine. Then, guys, we have one more disease. So this was Ehlers-Danlos syndrome. And the last one is epidermolysis bullosa. Right? This mostly affects a type 7 collagen. And epidermolysis bullosa, is a disease where there is a frequent appearance of blisters and fluid-filled vesicles on the skin. Right? So the skin is very weak in that case. That's the name. Epidermo means skin. Bullosa means formation of bullae, right? Vesicles. Okay, blisters. So these are some names of disorders that you must know at least to write in the exams, like enumerate the disorders of collagen synthesis. So scurvy and Menkes disease are also part of that. Fine, guys. Okay.